Hello. It is well said that purpose of education lies in its application. And so to accomplish this, we have arranged this series of webinars. Namaste and wish you all a very good morning. This is Rachna Joshi. I'm an assistant professor at School of Pharmaceutical Sciences at Mia University. And it's a great honor for me to extend a cordial welcome to all the dignitaries and faculty and student participants on behalf of School of Pharmaceutical Sciences, Faculty of Paramedical Sciences at Mia University. I welcome you all to this 1 week webinar series on connecting the classroom to industry. And the main objective is equipping the students with the real world experience. Dear participants, as per our tradition, we always seek blessings of Divine Supreme or Almighty before the commencement of any event. So let's we all be a part of this divine ritual. I request you all to be a part of the prayer. Varun. Again, I welcome you all uh, to this 1 week webinar series. Thank you, everyone. Now let us have the pleasure of listening to the opening remarks. May I now request the coordinator of this event, Dr. Shamiksa Patel, for the welcoming remarks.
Very good morning to each and everyone here with us for one week webinar series on connecting the classroom to industry, organized by School of Pharmaceutical Sciences, Faculty of Paramedical Sciences at New University. On divine presence of Hariprasad Swamiji, our president and mentor, Param Pujya Tyagvalla Swamiji, Principal Sir Dr. H.M. Tang, my fellow colleagues, all dear participants, it's the moment of extreme pleasure to welcome you all in one big capacity building webinar series. It's my great pleasure to welcome our respected and beloved speaker of today's talk, Mr. Nilesh Darji. This is a webinar series in which we should all deeply interested because academic professors and industrial professionals both have in-depth understanding and knowledge of principles and theories within their field. The distinct collaboration between industry and academia combine the best of both the worlds and are highly advantageous to students. Industrial professionals tend to have more comprehensive knowledge of inner working of the professional world, including the market, system, and process, so that connection with the industry experts allows students to focus on what they want to do in life, broaden their experience of prospective career fields, and help them on their future. So, student the connection between classroom learning and the real world. Within this thought in mind, we plan out this webinar series for UG and PG students, research scholars, and faculty members of pharmacy. One more thing I also want to share related to the keyword of our today's session, that validation. Validation was firstly apparent in 1978 United States by Food and Drug Administration to investigate failure of product in sterility. Now, it is very common concept in pharmaceutical industry to ensure that the process will produce consistent and repeatable results within their predetermined specification. So that every industry have the separate department of validation. We have received more than 350 participants, so that's my humble request to all the participants. Please be attentive and get knowledge about how classroom curriculum is applicable in industry. So thank you all of you. I again welcome you all for this wonderful and informative one week webinar series. Thank you all.
Thank you very much, Samiksha madam. Your remarkable and thought-provoking remark set a perfect platform for our speaker to deliver their presentation on connecting the classroom to industry. Now, let me introduce our today's speaker. I must say it's very huge and impressive biodata. Mr. Nilesh Darji. He is a professional and diligent personality with M.Pharm in pharmaceutical chemistry and having almost 10 years of experience in quality assurance division. He is having a great expertise in the field of data interpretation, instrument calibration, SOPs, documentation, GMP, GDP, QMS activity, and validation. He is excellent in ERP SAP, and in written and oral communication. He completed his diploma in packaging and development from Indian Institute of Packaging Development IIT Mumbai. He has been actively participating in various conferences, training programs, and presentations. He has a dual experience of working with academia as well as industry. He served as an assistant professor in PG Department of Pharmaceutical Chemistry for about a year. He has worked with Intas Pharmaceutical Limited as a senior officer in Quality Assurance Department. He has also worked as an executive in Quality Assurance Department in Emnil Pharmaceutical Private Limited. He has also worked as a senior executive in Quality Assurance Department in Concord Biotechnology Limited and in Equilife Healthcare Private Limited. Um, For the last 1 year, he has worked as a quality assurance calibration and validation coordinator at Jamifin Corporation, Brentford, Canada, and currently he is working with Novocol Septodone Pharmaceutical Limited as a validation specialist. Thank you so much, sir, for being present with us. Before I request sir to start his session, I would like to mention few general instructions for all the attendees. So there are certain things that we all are expected to follow during this webinar for the seamless webinar experience. The attendees will remain muted during the course of webinar. So we request all of you to take a note of your question or queries if you have any during the session. We will have a separate question answer session at the end of the webinar. So your queries and doubts will be addressed there. During the session, if any participants would face any kind of inconvenience or technical issue, you can let us know in the WhatsApp group that you all are connected to. Our volunteers will try to resolve your issue as quick as possible. After the webinar, everyone will receive a feedback form. The feedback form link will be shared in the chat box here and it will be also shared in the WhatsApp group. So it's mandatory for all the participants to fill the feedback form. The link will be open till the 30 minutes after the session. So you all are requested to fill up the feedback form in order to receive a certificate. Thank you so much. Now I request our today's um, expert, Mr. Nilesh Darji, to commence his session. Over to you, sir. Just wait for a few minutes. Sir are just about to start his session.
Start now.
Hello. Hi. Just a minute. No. Hi. Just a minute. Yes. So can you hear me? Okay. So probably I missed the introduction portion because of the connectivity issues. Uh, so let's directly start with the validation activities and our presentation. Let me share my screen. Can you see my screen? Can you see my screen? Anisha, can you see my screen? Okay. So our presentation scope regarding the validation. So what is going on currently in industry regarding the validation activities, along with the qualitative and quantitative applications in quality assurance department. So validation is one of the biggest topic in the pharmaceutical industry. So today's the topics are like QMS, what is the validation, what are the requirements for the FDA, like US FDA and other regulatory bodies, uh, equipment qualification, process validation, and facility, water system validation, computer system validation, and the cleaning validation. So what is the QMS activity and which QMS activity drives the validation and qualifications. So QMS means the quality management system, and quality management system has multiple different aspects for to drive the various activities in the pharmaceutical industries. And each company, each pharmaceutical industry has the quality programs. For example, like for any kind of the activity in the pharmaceutical industry, you must have to go through the qualitative program. Quality program has multiple events like change control, deviation. There are two types of the deviation: planned deviation and unplanned deviation, corrective actions and preventive actions. In short, it calls kappa. Third one is the like the non-compliance report and other activities, kind of the OOS, OT, APQR, and all that. But which activity drives the validation activity? First is the change control. So if you want to do any kind of the validation, any kind of the change, if you want to implement any kind of the procedure in pharmaceutical industry, you have to initiate the change control. Change control is the document which provides all the which represent your goal, which represent your scope, objective of your task that you want to improve in your company, in your organization. So change control has multiple names, like few companies say CCR, change control report, few companies say CCN, change control note, few says CCF, change control form. So all the terminology are different, but the work and like the process for the change control will remain same. So how validation will start? How do you think, like what is the importance of the validation? So if you want to implement or if you want to develop any new process in a pharmaceutical industry, if you want to design a new product in a and want to launch into the market, you have to go through the validation activities. So first of all, you have to initiate the change control. So change control will represent the validation related activities to the all cross-functional team, to the quality assurance department, to maintenance, to production, to quality control, QC, to everyone. That is how they will know that this new product will gonna come in our company and that will be launched to the market. So everyone will say their view, what are their requirements. So that is the main QMS activity in validation. So our project, our scope of that presentation is not the QMS activity. This is just brief intro regarding the QMS activity, but the validation is our main topic. So how validation governs all the activities? So first of all, if you want to decide, if you want to buy one equipment for any kind of the product, and then how will you decide that which equipment we need to buy, which equipment will fulfill our requirement? So first of all, each and every company has to prepare one URS. This is called this all package, URS, FAT, set, risk assessment, configuration specifications. Everything it's called the validation package. So for any kind of the validation activity, you will have this type of the document. First, you will generate URS. In that, you will say, this is our requirement. These are the functional parameter of the equipment. These are the acceptance criteria of our product. What will be the required for the microbial testing? What are the requirements for the chemistry testing? That all you will decide. You will tentatively say in URS. Then, for example, you want to buy one blister pack machine, for example, BQS machine. So you will generate a URS. In that, you will say these are the dimensions of our GMP area, and we want to pack 10,000 tablet or 10 lakh tablet in a per shift or per 8 hours, or like you will mention each and everything in your URS. This URS you will submit to the vendor. From whom you want to buy their equipment. They will review your URS and they will say that yes, with this type of equipment we can provide you and you can buy from us. And at that time they will say we will help you in installation qualification at this point, and after that you have to do the operational qualification and performance qualification at your end. So that is called the site acceptance test. Company, like the vendor, will come at your site. They will check against require URS. They will say yes, your URS can be fulfilled by us after checking your site. Then URS and site, and then you will go for the risk assessment. So what is the risk assessment? That is one of the most important topic in the pharma industry. For any kind of the activity, you will generate a risk. For risk assessment, you will go to the customer level. You will think like what will gonna happen if product or equipment will fail for its intended purpose. At that point, you will decide low risk, high risk, and moderate risk. So if equipment has a low risk, if something went fail, if blister pack machine failed for the sealing operation, then what gonna happen? The product, your content of your product will expose to the environmental condition, external environment. That will impact, that will lead to the product cross contamination, product contamination, and that will impact on the patient's health. So based on the risk assessment, you will decide how often you need to revalidate your product, how often you need to revalidate your equipment. That is the main intent of the risk assessment. And once you have the risk assessment, you decide like this is the risk, and at that time you will identify the project level validation. In project level validation, you will decide this type of the testing we need to do, we need to perform, we need to double check, we need to challenge this type of the testing for the particular equipment, either for the particular product. Configuration specifications mainly used in the computer system validation. Before you launch any kind of the software in a pharmaceutical industry, you have to decide its configuration. Configuration may decide user level access. For example, like operator has only few access. They only can run the machine. They can start and stop the machine. They cannot change any kind of the parameter. Supervisor and manager level has access for the review. This and admin level can alter all the type of the parameter. They can change anything user. They can generate a user, new user. They can delete user, and they can have the access to the audit trail. So what is audit trail? So audit trail means any kind of the activity you perform within the software, it will be stored at one location. Whenever regulatory agencies, means auditors, for example, FDA, either MHRA, either Brazil, Anvisa, WHO, whoever will come, they will ask you to download the audit trail. They will go back there and they will check what you have done before starting the operation. If you, and in pharmaceutical industry, you cannot delete anything. You cannot, you just need to, you can archive, but you cannot delete. If you did something wrong, you have to keep everything over there. Auditor will see and they will look how you resolve the issue. Then factory acceptance test. In factory acceptance test, you, once after your equipment is totally designed at vendor site, few of the representative from our company will go to their plant. They will check the equipment against their URS. Like, does this equipment has the same features, same requirement, same acceptance criteria, what we want? If you are satisfied at that point, you can say them, you guys can come on our site, you can install the machine. And after the installation, you will perform the validation and qualification activity and this periodic review. that is what I said. In risk assessment, you will decide at how often we need to revalidate the process or equipment. So either may be like annual revalidation or biannual or 5 year. Like if you have the high risk to the product or to the equipment, you need to revalidate the same equipment at every year. And regulatory bodies always want to see how you design your risk and how often you are revalidating your system. So the basic terminology of the validation in FDA. FDA means US FDA, Food and Drug Administration. Everyone is going to follow the US FDA guideline for the validation activity. Most of the guidelines, like the chemistry, micro validation, QA, technical services, corporate engineering, everyone is following the FDA guideline to perform the validation activity. Validation is establish a documented evidence that provides a high degree of assurance that the specific process or equipment will consistently produce a product and results meeting its predetermined specifications and attributes. Once you validate your system, why you are validating? You already decided your acceptance criteria. You know what are the requirements from the regulatory bodies, what are the requirements for the particular API assay, percent assay, that you are already decided in URS. Now you are doing the validation. At that point, you will say, we validated 3 batch at the initial, before we launch into the market, we validated 3 batches and we found the consistent result. And based on that, we will gonna follow the same procedure, and that will remain consistent throughout the product life cycle. Either it will be 10 years, 20 years, whatever. So that is about the product. In equipment should have to perform as intended. Like if you want to seal the product, if you want to seal the plastic bottle at 120 degrees centigrade, then today you seal at 120 degree centigrade, and like after a few days, if you seal at 120, the product should not be get degraded. Product should remain same. Its efficacy and its intended purpose should remain same. The percentage should not be should be lower than what is expected. So that is the main reason why we are doing the validation, to identify. Once you do the validation, you have to generate multiple documents to follow throughout the product life cycle. Either batch record. Batch record means some companies say that BMR, batch manufacturing record. Few companies say that batch BPCR or BMCR. So like batch manufacturing control record, something like each and every company has a different terminology for the batch records or SOP and all that. But once after the validation, you will identify what needs to be done during the routine process. So you generate a batch record or master equipment file in master, like batch record for the product and master equipment file for the equipment. So let's see installation qualification. So in installation qualification, it is very simple. In IQ, you don't need to be worried. It should be installed as per the manufacturer's specification. They say you should operate the machine at this speed, so you have to operate at that speed, not more than that. Otherwise, like the multiple maintenance and there will be more and more breakdown, and you cannot run the machine consistently. In operational qualification, like, so in OQ, machine has 1,000 kind of the functional parameter, 1,000 kind of the facility you can use like multiple way, but you have to decide what is our requirement that you already mentioned in URS. So for example, like you said, my machine should operate at 120 speed or 120 degree temperature. Then you should qualify machine at 120. You can qualify at 3 different temperature, 115, 120 and 125. Then you define the range, like we need to operate the machine in between 115 and 125, but your requirement is 120. So that is what you're going to identify in your operational qualification. You said in URS, we need, this is our operational parameter. Then your machine should be operated at this particular point. In PQ, performance qualification. In performance qualification, you're going to identify its consistency. Like you operate machine 24/7, like for a week. It should give you the same performance. It should not automatically reduce its speed, its temperature, its selling quality, not like that. So IQ, OQ, PQ, few companies and few equipment do IQ and PQ all together. Sometimes vendor provide you will say that we'll do the IQ, OQ, and PQ. For example, if you buy HPLC from Agilent or Water System, like the name of the companies who generally provides the HPLC and all that, they will come at your site. They will install. They will do the operational qualification. They will do the performance qualification. But they follow the general criteria. They follow the general performance qualification. But your company, you have few product, like you have controlled substance product, you have anesthesia, and you will have very like tough parameter, like not broad range for the acceptance criteria. But vendor didn't do that particular test, so you need to prepare a separate performance qualification protocol. You need to do a PQ at your end, not by the vendor end. So it depends what are your requirements. That is why the URS is one of the most important subject and one of the most important document. If something you are not following in URS, then it will be a deviation. So that is a part of the QMS activity. I will tell you something regarding the deviation, what are the planned deviation and unplanned deviation at the end of this presentation. So in FDA, like if you will, like any of the first like student will go in industry, they will definitely gonna ask you what is FDA. So FDA is the Food and Drug Administration. It has a CFR. CFR is the written document. It's a huge document. It has different part, but generally we need these two parts first to get enter into the GMP area. So 21 CFR part 11. There are 21 means 21 chapters. CFR means Code of Federal Regulations and part 210. There are multiple parts in each chapter. So 21 CFR part 210, that will give you the whole idea about the like good manufacturing practice, processing, its packaging and holding of drugs, means API, vehicles. But part 211 will say about the finished product. Like once you in a manufacturing unit, you will prepare a tablet. That's not finished product. That's your drug. If you once you pack them in a blister, you pack them in a like carton, then in a corrugated box, that's your finished product. End product at the end, like which is ready, which is good to go to the market directly. Once quality assurance team release the product, it's good to go to the market. That's called the finished product. Tablet is not the finished product. So this 211 will give you idea about the finished product, and 210 will give you the idea about the drug products, either API, vehicles, whatever. That covers carton, blister foil, plastic bottles, cap, everything. And these are the links from where you can download the. I will share this presentation with Samiksha. She will give you this slide, and you can download the guidelines and you can go through it. So our main focus is the process validation. So how do you know that like we need to launch this product into the market? There are two departments in pharma industry. One is the R&D. So R&D will search, they will look into which product's patent is going to be expired. They will start development into that particular product. They will develop at lab scale, then they will go for the pilot scale, and then they will go for the exhibit batch. That exhibit batch will go on and submit to the FDA or either any of the regulatory bodies. Once they approve your exhibit batch means the like minimum batch size. For example, you will perform a 10,000 tablet exhibit batch, and as per the regulatory guidelines, if you have 10,000 tablets, batch size is the 10,000 tablet, then you can manufacture 10 times of the 10,000 and you can sell to the market. And at that time, you need to go for the process validation. So process validation is always a product specific. Each product has a different criteria. So few products are light sensitive, few products are chewable products. Chewable products are like Montelukast. Is moisture sensitive. You should have controlled environment. You're like in pharma industry according to zone A, zone B, and zone A to C, A to D. There are different environmental conditions. So in pharma industry, you have to work in a controlled environment where the temperature limit is 25 plus or minus 2 and humidity 55 plus or minus 5%. So if your GMP area doesn't have that required temperature and humidity, you cannot start any kind of the activity. You cannot even start weighing of the API. You cannot start dispensing activity like granulation, compression, coating, imprinting, even packaging. You cannot start that. But the product, which are generally light sensitive, you should pack into the sodium light under the sodium light lamp. You cannot pack in the normal light for Montelukast kind of the tablet. There are only few tablets which are the like moisture sensitive. So particular room has a humidity of 25 plus or minus 5%, not 55. So it's a product specific. So if you don't follow the environmental condition, your product will gonna be degrade before it goes for the tasting. So that is how the process validation will work. And revalidation, there are two terminology: revalidation and the requalification. So revalidation is a routine exercise. That is what I said. Once you generate the risk assessment, you decide that the product and the equipment has a high risk. You have to validate like either every 6 months or revalidate either every 6 months or annually. And at that time, you have to compare your revalidation activity to the original qualification, original validation activity. If something is different with your original validation, then you have to think like you have to redesign your product or you have to improve in case of the equipment, you need to improve the frequency of the preventive maintenance. Like if you are doing the preventive maintenance of the equipment every 3 months, then you have to start doing every month. But if everything is good to go, like no, like your revalidation parameter and the original qualification parameter are exactly same, then you can increase. You can increase like 3 month preventive maintenance to 6 months. And same in the case of the uh, calibration. If we need to do a calibration of our equipment for weighing balance, like if you buy weighing balance, you do the qualification, it's like every month, every annual calibration is good. Then you can increase your qualification calibration frequency. Like you can go for 1.5 year, like step by step. You cannot double that calibration frequency, like what annually to directly by annual. You cannot go like that. But that is what it decide. The revalidation will decide its consistency and what you need to do to improvise the process or the equipment quality. Whereas the requalification, for example, you have one product. So you are using a API from Dishman Pharma, and there is something wrong with the Dishman Pharma API. Okay, so and FDA will give you the notification that you from now you cannot use the API from Dishman Pharma because this is not as intended or this is not as per the regulatory requirement. So what will you do? You will find another vendor who is preparing this type of the API. So now you change the API. You don't know how that different company is manufacturing that API. So you have to do revalidation, requalification. So at that time, it's almost same as the original validation. You have to follow each and every process from the beginning, and you need to requalify your process, either equipment. In case of equipment, you are changing a critical part of equipment. The part might be a like for like. You bought the same part like from same model, same company, and everything is good to go. Then you don't need to do the whole requalification. You just need to do the re-verification at that point. But it must be like to like. Like to like means exactly same model, same company, same manufacturing, everything, dimension, parameter, each and everything should remain same. But sometimes you like that particular vendor stop that part, like the particular part manufacturing. They improvise that particular part. What you will do? You need to go for the requalification. You cannot re-verify. That is called the requalification. So uh, in process validation, there are three types: prospective validation, concurrent validation, and retrospective validation. In simple terminology, prospective validation used for the new product. But in a prospective validation, you before you go for the validation, you have to decide how many batch you will choose for the validation. And each and every regulatory bodies expect minimum 3 batches for the process validation. So if you are going to do a 3 or 5 validation, prospective validation batches, you cannot sell any of the batch. You have to complete all 5 batch. You have to prepare the final report of that all 5 batch, and then you can sell to the market. So that is only for the new products. Concurrent validation means you are changing a parameter. For example, like you are changing the assay limit, you are changing the pH of the final product or the final tablet, like either you are going to change the hardness of the tablet of your existing product, then you can go for the concurrent validation. But regulatory bodies wants to have 3 minimum concurrent validation batches for even existing product. But sometimes you don't want to do, you know that like it's a minor change, it's not major change. So but you have to prove that it's not major, it's a minor change. This is not going to impact on the product quality and ultimately to the customer. So at that point, you have to generate the risk assessment. Based on the risk assessment, you will say that this product, this change has minor change. What are the minor change? For example, you are buying one API from X company, but at some point you cannot go for the, like you are buying through a distributor, but that distributor stopped the selling their product. So you need to identify different distributor to buy the product from the manufacturing company. So you are altering the distributor, not API, not manufacturing company of the API. That's minor change, but you have to prove that this has a minor risk. And retrospective validation, this is no regulatory body is accepting this type of the validation nowadays. No one wants to see the historical validation. Like in this one, what you gonna do? You will choose, like you have X product, you want to do a retrospective validation. You want what you gonna do? You will in year of 2021, you manufactured like 13 batch of that X product. So you will collect each and every data, QC data, microbial data, production data, and you will compile in a single report. And based on that is called the retrospective validation. But no, for sure, no regulatory body expect the retrospective validation. That is just for your company internal identification. So these are the three major difference in the prospective validation, concurrent validation, and retrospective validation. So let me provide you a sample protocol for the process validation. So uh, in process validation, how you generate the protocol, it's critical because once you mention something in the protocol, you have to follow. If you once your protocol, so these are the approval pages. So here here the author will sign, reviewer, and approval. Approval might be generally in process validation, approval will be first will be the production, one will be the QC, one will be the micro, one will be the validation manager, and final from the quality assurance manager. Generally, these are the main 5 topics that should be into the validation protocol. First of all, introduction, validation approach, responsibilities. We'll discuss later on. So what you gonna do in introduction? In introduction, you have to mention the product batch size. First of all, the product name. So you have to be very specific to your product. You have to mention the name of the product which is exactly gonna be launched to the market. So, for example, this is the diazepam injectable solution. It's a volume. Either you can say volume, like 10 milligram per 2 ml strength, or volume, whatever you refer at your end. Like but different company has a different terminology. Either they can strength or something, whatever. But you have to mention the batch size. This is one of the most important things. If you don't mention the batch size, you cannot manufacture the whatever batch size you want, because at particular batch size you have particular identified equipment, particular identified like containers, vessels, and everything. So, and where you wanna manufacture. For example, you have the same company has 2 plants. For example, like Intas, I'm just giving the example Intas because Intas has a plant in Matoda, and another plant is in a says pharma says. Both are like just 1 kilometer away from each other. So they cannot validate batch at Intas Matoda plant and then they can start the manufacturing at Intas says plant. No, that is not possible. You have to validate your product at the particular location. You cannot transfer your method. If you want to transfer a method, you have to do at least minimum of 1 validation batch at the new site. So other thing, you also have to mention the major equipment which you want to use for the manufacturing of that particular bulk solution, either or a tablet. This protocol is related to the liquid injectable. But if you have tablet, then you have to mention the ID number of the particular equipment. This coater, this compression machine, this granulation machine, either capsulation machine, imprinting machine will gonna be used for that particular tablet or particular solution. Also, you have to mention the room number. Each company has company layout, and they gave their identical number to the company. So why we need the name of that room? Because each particular room has grade A, grade B, grade C type of the grades. You already studied in your educational career, but that is the reason why you need to mention the room number different. Like we have 2 different rooms because in this particular process, 2 different API will be manufactured in 2 different rooms and then they're gonna be mixed up in a separate room. So if you have different unit, then this introduction will give you the whole information about your product, what is the name of the product, what is the strength, batch size, which major equipment you're gonna use, where you going to be manufactured, and particularly at that location in which room you gonna manufacture that particular product. And here these are the sterilizer, washer, depyrogenation tunnel. There are the major equipment that gonna be used for this particular product. And these are the utilities. So HVAC system provide you the differential pressure. So for example, if you have seen the GMP environment, there are 3 rooms. Always there will be 3 rooms. So first is the gowning room, another one is the secondary room, and third one is the primary room. So primary room where you gonna manufacture, you gonna compound, you gonna compress your product. So that room has the negative pressure. Have to have the negative pressure. Then that of the secondary room, and then that of the change room. And these all 3 differential pressures should be relevant to the corridor. Why? Because you have one GMP room, another room is next to the your GMP room. So X room has a Y product and Z room has a product. So if you don't if we don't monitor, or if we don't keep controlled differential pressure, then during entry and exit of the manpower or material, the product powder gonna comes outside, and the product contamination, cross contamination might be possible. That is the main reason why we need the HVAC system. HVAC system is always maintain the differential pressure between the adjacent room to prevent the cross contamination of the product. Then the WFI. Each company has a WFI plant, and the WFI plant, this is gonna be tested every day by the micro department for bioburden and endotoxin test. This is the pure steam. Few product needs steam, so you have to identify which type of the steam you're gonna use, pure steam or plant steam. So some of the product have uh, like chemical reaction, so you have to provide the nitrogen environment. You all know that like nitrogen is the inert gas, so you might need to provide the nitrogen gas to prevent the chemical reaction in between the product. For example, there is a one product, what's the name of that product? So what there was one of the big issue in that product. Like once you pack the tablet in the blister, there will be the bulging issue, because there was a reaction in between the 2 API, and the tablet will start the bulging, and blister is automatically gonna like blow up. It's totally like becomes unsealed within a 1 month of the manufacturing. So in that cases, you to prevent the chemical reaction in between the API, you need to provide the nitrogen environment. Compressed air you need for mostly for the equipment. Sometimes you some equipment has sensor. So if you want to stop one function of the equipment, you should use the compressed air. So once the sensor sense that your this activity is done, you want to stop that activity, then compressed air will give the command to that particular soft sensor, and that activity will stop, and next activity will gonna start. So this is Yokogawa. Like Yokogawa is the name of the software, which is 24/7 by 365 days gonna monitor the differential pressure, environmental condition like humidity, temperature, nitrogen gas pressure, compressed air pressure, everything. Like this, anytime you will log in, for each 3 seconds you will get the results. So if something is out of spec, like temperature is not in control, you will get alarm, and your machine automatically will stop. It will not work unless and until you will go back to the normal operational parameter. So these all things you have to mention before proceeding for the process validation. Here is the product detail, and generally each and every process validation had 2 parts. For tablet, it's manufacturing. For liquid, it's compounding, and second portion is the packaging. So In next section, you will say validation approach. So how will you do the validation? You have to mention what are the critical parameters, what you want to do in your validation. So at that point, you will say the batch compounding and its product transfer. Means in manufacturing unit, you manufacture the tablet and either liquid solution, injectable solution. Then you have to transfer to the packaging department for the finished product. So for during the transfer, what precaution you need to take, that is most important, because at this point, at compounding manufacturing point, you did a tasting. SA was okay, every pH was okay, everything was okay, but there might be a chance for the product contamination during the product transfer. Then it's primary packaging and its filling process. And these are the cartridges. This product is going to be packed into cartridges. So the cartridges inspection. So if you have tablet, then blister inspection, like HMHTP bottle inspection, capsule inspection, like what type of the blister you have, like aluminum foil and like alu pack, aluminum pack, or like aluminum or paper foil. There are different types of the packaging for the each product. So that is you here you have to identify what you gonna taste it, how you will do the validation, and you need to mention the number of the validation batch here. So sometimes in concurrent validation, as I said, you need, if you are going to do a 3 validation batch and you want to sell one batch into the market, then you need to prepare a one interim report. Once that one validation batch is done, you can prepare interim report, you can sell to the market. Second batch, prepare interim report, can sell into the market, but not in case of the process validation. And at the end of all 3 batches, you have to prepare the final report. And these are the responsibilities. So this is one of the most important. You have to say what type of the validation, which department will perform what type of the responsibility. Main responsibilities of the validation department. They will ask all cross-functional team to help them as required to operate the machine, because validation personnel will not gonna operate the machine. He's not gonna compound the product. He will give you the idea. He will give you the procedure. But the work has to be done by the production department, because they are the people who are going to do the same activity on routine basis. And QC, so QC has 2 departments, micro and QC, chemistry. So QC will do their testing and micro will do their testing. QA will be the responsible for overall activity of the validation. They will QA work as a police. They will say yes, you are good to go, then you are good to go. They will stop. They will say stop, then you have to stop. So here you have to identify the quality CQA parameter. In any kind of the validation activity, any regulatory body will first ask you the question about the CQA, what CQA parameter you have identified. So here, these are the particular parameter, CQA parameter of that particular product, diazepam. But at bulk solution, either bulk solution, you can say bulk solution, you can say like blend, common blend for tablet, bulk solution is for the injectable. Then for solution, it's filtration, and for tablet, it's a granulation, and at the end of the operation, like finished product. So at that time, you will say, we will test the assay of the particular API, how many different type of the API you have, and like at each processing step, you will identify each required acceptance criteria, which you already mentioned in your URS. So again, you will here mention that all the equipment, system, process. Okay. So what type of the equipment and utilities you are going to use you have to give its a raw material number. Water for injection has different type of numbers, because in many plant, like if you have the big plant, then and you have multiple department, then you will always decide from this unit the WFI will be supplied from particular WFI plant. So if you will not identify which WFI you want to use, it will be a troublemaking during the validation operation. So you have to identify each and every parameter: compounding vessel, tank, like filling machine, blister machine, granulation machine, coating, inspection, everything. So this is the product specific, but this will give you the idea what you need to mention during the protocol generation. Here, as I said, there will be two parts. Like this is the compounding, and the other one will be the particularly packaging. So here, before you go for the validation, you will have the master batch record. Okay, but this master batch record you can change once after the validation activity. At the end of the validation activity, you say you identify there is a gap in the batch, like this should be that, but it is not mentioned in the batch record for the routine operation. Then you can implement the master batch record, and this master batch record will remain with the QA. They will issue to production department every time they wants to manufacture the product. And at that point, you will say what are the acceptance criteria, particularly for that compounding operation. So the process validation test results must conform to the specific acceptance criteria. So what are that specific acceptance criteria? That is a certificate of analysis, COA. That is responsibility of the chemistry department, quality control, QC department. They, before you go for the validation, based on the vendor certificate and based on the FDA's requirement, QC department or either R&D department will prepare one COA. At that point, they will say the X product, X API, SS should remain in between this range, pH should be remain at this, and like either you need to do what type of the test, IR, UV, HPLC, GC, what type of the test you need to do. That will be decided by the R&D department, either QC department. Because QC is a like it's a different terminology. Like QC has two type of the department, like one for the commercial market and one for the analytical market. So analytical, that will gonna help to the R&D department, and commercial QC will help during the routine operation. They do the routine testing for the product which is already into the market, not at the stage of the validation. So here particularly you will again mention the equipment and product detail, particularly for the compounding or manufacturing portion. And this here you are seeing that this data sheet, this data sheet will comes later on, because you will understand later on, because I cannot explain at this point without showing that data sheets. But for the compounding and transferring process verification, you have to prepare one flow chart. How you will do? As I said, like there are two different API. They're gonna manufacture at two different room, and then finally they will be mixed into the third room. So this is how this diazepam, benzyl alcohol, sodium benzoate, and W5 will be manufactured here. These other APIs will be manufactured here and will be combined into the third room. And at that point, you have to do the like whatever type of the adjustment. Like sometimes you have to mix the temperature at particular speed, mixing speed. Sometimes you have to maintain the particular temperature. So that is like as I said, sometimes you need to provide sodium light. Sometimes you need to provide a low humidity or higher humidity. So that is what here you have to show. And this is the same. These all are there. This is all we already decided. Like we already discussed. This all pH measurement, visual inspection, analytical testing, this all will come from the certificate of analysis, which is prepared by chemistry and QA R&D department. So you have to you do the testing for the color, clarity, pH, ethanol, like what type of the assay you need to do, and all that will stay here. So here at the end of the compounding, that means that is called the in-process testing, because you are done with your tablet manufacturing, either injectable solution manufacturing. So you need to do the microbial testing as well as the chemistry testing. So this assay are the chemistry testing, QC testing, and these are the microbial testing that over there. So here you have to identify your sampling plan. So you have 150-liter batch. I think you might remember that the introduction section we said 150-liter tank will use tank 34. So you need to collect the sample from the different height of the tank. So one sample will be collected the top of the vessel, middle of the vessel, and bottom of the vessel. So one sample of 250 ml and one sample of 50 ml. So this 15 ml is for the microbial endotoxin test, and this one is for the bioburden test. From middle, same, and from bottom, same. These all 6 sample, like these 3, top, middle, bottom, for bioburden, their results should be consistent. Like there should be, I think you know that how to find out the R square value. Based on that, that is already mentioned into the C of A. So that should match in between them. So here, in this case, this is the process plan. This is whole process, how we will do the operation of the manufacturing, starting from the dispensing to the compounding, then transfer, and then filling operation. This is the whole process flow. And after doing the compounding, you will go for the filling operation. And after doing the filling, you will go for the same testing. So now at the end of the after compounding, you will go for the product transfer to the packaging department, and before filling, they will do the more testing. So at the start, like before start of the filling, they will collect the 200 cartridges, middle of the fill and end of the fill. They will collect these cartridges and they will go for the testing for the same, for the microbial endotoxin and microbial bioburden. So ultimately they are testing the same product, but at each and every step. So this one, now let's see the appendices. So this appendices is a critical instrument and instrument calibration. So during compounding, you need to use many different types of the instrument like pH meter, weighing balance, and like thermometer and all that. So whatever thermometer instrument you use, you have to mention here its name, its calibration. It should be a calibrated. It should be in calibrated state, and this is the responsibility of the operator, of the compounding person. Like before they start using of any of the instrument, they have to have the instrument in calibration state. So, and these are the raw material spec. This raw material is already released before they launch to the market, before they launch for the manufacturing. Sorry. So these all are released. The certificate of analysis for the this raw material from the chemistry department as well as the vendor, whatever raw material or API or vehicle, whatever vendor will supply you, they will provide you the certificate of analysis that our product is good to go. It's good, it falls the regulatory criteria, but you are not going to trust them. You have to do testing at your site too, and then you will release for the production use. So this is also you have to mention for the in validation. And these are the raw material number. These are the like biodata of the raw material which you are using. Its manufacturing lot number, and this is also done by the operator. Then component specification. So what type of the packaging component you are using, like the blister foil, capsules, like what type of the capsule cell you are using. Then what type of the cartridge is HMHTB bottle, like what HMHTB bottle you are going to use for the different size, like 30 count in one bottle you will have 30 tablet, 60, 90, 500. So each item, each packaging material has its own code. So that is what you have to mention here, and this will be provided by warehouse department. But it is the responsibility of the production to mention here. Then this is the like datasheet 825 is your operation, manufacturing operation from the start. So whatever you mentioned in your objective in your introduction, you have to mention again. It's room class, which room you are going to use, product name, what batch manufacturing record you are using, everything you have to mention here. And this is the compounding process. Like that is you will going to collect this compounding process from the master batch record. So this might be a right or this might be a wrong. We are not sure because we develop the product at lab scale, not at the at production level, at pilot scale. Okay. So we don't know how it works. So once, but if anything failed from this mentioned parameter, for example, like here you are saying that yes. So here you are saying that quantity of ethanol 96% added and pH record. Here there are no specific criteria for the pH. But if you will say like pH will, yes, here you say that record pH, its target pH is 6.4, but its range is 6.3 to 6.5. But if it is like 6.2, then it's a deviation. And you have to separately investigate why the pH is not within the acceptance criteria. And you have to identify the root cause. You need to identify the corrective action and you need to identify the preventive action. That is called the kappa. So if something failed, there must be some root cause. And if you identify root cause, then how will you correct that mistake? That's called the corrective action. And how will you prevent that activity to prevent its reoccurrence in future? That's called the preventive activity. So you will identify one permanent way that this will not going to happen again in future. So that's called the preventive activity. Then this is all like the compounding process, and these are the acceptance criteria for the chemistry. Once you identify your product at like compounding bulk level, at filling level, and at finished product, this acceptance criteria, this assay acceptance criteria is the same, will remain same. They are not going to change, but it should be within the acceptance criteria. If something failed in between, then you have to go for the deviation. The many deviation has many parts, like the minor deviation, major deviation, critical deviation, and each minor, major, and critical has different corrective action. But if it is a critical, then in 99% case you have to scrap the batch, because the critical deviation is a like fatal impact on the customer. So critical means mix up of the product. You mix two product. Either you used expired raw material, API has expired, no one checked it, you used for the compounding, that's critical. So at that point you have to scrap the product. You don't have any option. So that's called the critical deviation. This is for the, so these are the assay limits, and these are for the chemist microbiology. So what will be the bioburden? What will be the endotoxin, bacterial endotoxin results? What are the its specification? These specifications are already mentioned in USP for particular product, and you can refer 21 CFR part 210 and 211 for the finished product acceptance criteria. So here you are saying that, let me explain this one. For example, acceptance criteria, all sample meet the predefined acceptance microbial specification. If yes, then you're good to go. But if fail, then deviation, you have to go for, you have to click deviation, yes. Result, fail. And then you will mention the deviation number, like this deviation is generated for the investigation of this particular failure. Then that was for the compounding process, and this will remain same for the filling and product transfer. The same acceptance criteria will be there for the chemistry, because as I showed you, like we need to do the testing before the filling stage. Start of the fill, middle of the fill, and at the end of the fill. Same for the chemistry, same for the microbial testing. And at the end, if deviation, so there are two types of the deviation: planned deviation and unplanned deviation. Planned deviation means, what is deviation? Deviation means you are deviating from the standard operating procedure. So in a planned deviation, you know that like this is not our process, but whatever we are doing, whatever we are deviating, that's acceptable. You are going to justify before you are doing the act. But in unplanned deviation, someone did a mistake. It's a failure. That's called the unplanned deviation. And unplanned deviation must have to be investigated. Planned deviation, you already investigated. You already know that we are doing which is not mentioned in our procedure. So you don't need to go for the investigation. And each deviation will have the corrective action and preventive action. If something failed, then you will identify how to correct that mistake and how you will prevent. That's a QMS activity. And even in unplanned deviation, minor deviation will be like, it's okay, like it was the mistake was done by the chemist. He prepared a like wrong sample solution. So your corrective action will be like, we'll do the retesting, we'll prepare a fresh sample, we'll do the retesting, and product will be released. What is your preventive action? You will say that we will retrain the chemist that he is not gonna do the same mistake again. So that's for the minor. In major will be the failure to your acceptance criteria. So that is going to be investigated, and you identify the root cause, either repackaging or either scrap of the product or like pH adjustment. If pH is not will be not there, then you will decide like, let's readjust the pH and retest the whole product. So that is the my major uh, deviation. So also you have to mention all the applicable standard operating procedure, like this all SOPs will be used for the routine operation, and also we used to prepare the protocol. And this is a different thing, like this is, we are working for one like client, so that's their approval. So validation, this is all about the process validation. So process validation, like validation is a really big department. Like you cannot learn within a 1 hour or half an hour. It takes like whole life. For me, like I'm working in validation like since last 10 years, but I'm still learning, because each equipment, each product has a separate parameter and separate acceptance criteria. Because every like day by day new equipment are designs are coming there. So each and every equipment will have the separate functional parameter. You need to test them according to their manufacturer specifications and all that. And these all are the facility, like this is facility validation, what facility qualification and water system qualification. It's a vast, and I'm not going to cover this one here, but in facility, just let me give you the example. Like as I said, HVAC system, water system. So HVAC system, you have to do the testing for the HVAC system annually. You have to qualify the HVAC system before you start the HVAC, because that is the one who is going to maintain the differential pressure between the adjacent room. And how you will identify like your differential pressure is maintained within the adjacent room, you need to do the smoke study. So there is a one equipment instrument you can call that generate smoke, and the direction of the smoke should be from primary room to the corridor. That is, you have to do the annually. You have to prove that yes, our differential pressure is in correct direction. There are no chances for the product contamination. So facility means anything. Like if you block something, if you put a hole in GMP area, you need to do the requalification, because even a single hole will impact into the whole differential pressure. So it's a big like you need to qualify the warehouse. You need to identify 4 different spots for the temperature monitoring. Or like here in Canada, you need to identify 4 spots: hot spot for winter and summer, cold spot for winter and summer, and 24 by 365 days you have to monitor the environmental condition minimum at that 4 spot, because your warehouse is the place where you're gonna store all the products, and each product might be impacted by the temperature variation, humidity variation. So that is also a part of the facility validation. Computer system validation is one of the like it's a separate department from the validation. And cleaning validation. So whatever instrument you are going to use in cleaning validation, sorry, for product manufacturing, you need to identify that once you clean the equipment with WFI, that particular vessel, equipment, either even screw is not carrying your product. So that maybe you might know about the MACO test, maximum allowable carryover test in chemistry, like generally in QA during their like testing, you might know. So you have to identify the MACO value, maximum allowable carryover. So each regulatory bodies identified the maximum allowable MACO value for particular product, particular API. That during cleaning validation, you have to challenge that MACO value of that particular product. And for that, you have to do the bulk hole study. So, for example, your product manufacturing is done. So, what you're gonna do? You will keep your equipment dirty for 24 hours, 48 hours, 72 hours, whatever, like whatever bulk hold time you want to do. So you will keep your equipment dirty for particular time frame, and then you will clean it, and then you will go for the tasting. And it should not, like the particular product should not be more than that of the particular MACO value of their product. So that is called the cleaning validation. Like that is called the bulk product hold time. A bulk product hold time and cleaning validation both are same. Like both are relevant. If you want to do cleaning validation, you have to go through the product hold time, and if you want to do bulk hold time, you need to perform the cleaning validation. These are interrelevant. That's it. Do you guys have any question?
Thank you so much, sir. and it was really wonderful session. Thank you so much for giving in-depth and in detail explanation about various aspects of validation. Thank you so much. Um, Now we are having a question answer session. So if anyone from our participants is having any doubt or query, you can raise your hand and we'll give the unmuting rights to you. You can also write your queries in the chat box as well. Anyone is having any question, you can ask by unmuting yourself or you can write in the chat box. Okay, you can share your queries in the WhatsApp group as well. No one is having any question. We are concluding the session over here, sir. And it will be a great pleasure for us to listen to you in the future in our events also. Sir, you are not audible now. Hello? Your voice is not coming.
Can you hear me now?
Yes, yes, yes.
Yeah. So no problem. Like if anyone has any queries and like, if anyone wants to ask, like they can reach out to my my contact on my email address. I'm gonna share this presentation to Samiksha, and she can share this presentation to our team.
Okay.
And whoever wants to ask any question, they can reach out to me by email, or like she can give you the contact number as well. You can add me in a WhatsApp group so I can answer in the WhatsApp group.
Sir, there is one participant who has raised his hand. So we just ask them if they have any query or okay. Umesh Bhagwan Bhai, the person who is connected with this name, is now unmuted. So you can ask your question. You have raised your hand. Do you have any query? Okay, if you don't have any query, you can let us know in the chat box so we can proceed further. Okay, we are not getting any response. Thank you so much for your kind presence and for sharing all your valuable knowledge with us. Thank you.
It's my pleasure. It's 12 o'clock here, midnight. So I like to share my knowledge. I know it's validation is vast subject and it's not easy to give the whole detail within 1 hour. But anyone wants to know more about the validation, they can reach out to me. Either even for the QMS activity, for the calibration activity, they can reach out to me personally, and I will try my best to explain them.
Yes, sir. It's a vast topic, but then too, your attempt was really nice, and most of the things were covered in the session. So our students will definitely, or the participants will definitely get the nice insight regarding the subject. Just one more thing I would like to say. Sir, please don't leave the meeting as you are the co-host. So once the settings will be done, then we'll inform you to leave the meeting. Just a few
No
minutes.
problem. No problem. No problem. I'm okay. I can wait. No problem.
Okay. And we are sharing the feedback form link in the chat box, as well as we are simultaneously sharing the same link in your WhatsApp group also. So the participants are now informed to fill up the feedback form. The link will be open till 11 o'clock. So you all are requested to fill up the form as quick as possible. Thank you so much, sir. Once again, you may leave now, sir. Um,
Thank you. Bye-bye.
thank you. Thank you so much. Um, I request all the participants that we are concluding the session here, and we'll have our next session tomorrow at 2 p.m. in afternoon. So make sure you join on the time. Thank you so much. Have a nice time ahead.